Hi, today we are speaking with Dr. D'Alessandro about a recent publication in Brain Communications entitled Biallelic CRELD1 variants cause severe muscle weakness and infantile epilepsy. Hi Manuela, could you briefly explain us what your work is about?
Yes, sure. Our study identified two variants in the gene CRELD1 in a child affected by congenital contractures, severe muscle weakness, neurodevelopmental delay, and epilepsy. By introducing these variants into the model organisms Neurodevelopmental elegans, we will show that they are pathogenic.
And can you tell us what is CRELD1 protein?
CRELD1 is an endoplasmic reticulum protein that promotes the maturation of several substrates, including acetylcholine receptors, which are important for many processes like muscle contraction and neuromodulation. In C. elegans, animals lacking CRELD1 are still viable, while in mice, complete loss of CRELD1 is embryonic lethal due to heart development defects.
And can you tell us more about the variant found in your patient?
The biallelic variants found in the patient are a point mutation of a conserved cysteine in a key domain and a premature stop that removed the anchoring part of the protein. Because CRELD1 is highly conserved, we were able to test the same variants in C. elegans.
And why did you use C. elegans to study these human variants?
Our team, led by Jean-Louis Bessereau, has over 25 years of experience using C. elegans to study synaptic function. This model allows rapid genetic editing, functional assays, in vivo imaging of the entire organism, making it ideal to test whether patient variants affect protein function.
And how did your team come to characterize a CRELD1 related disorder?
This project started as a collaboration with Dr. Zeba-Atin Sayrak, who was treating the child. He sequenced the child's genome and identified two different variants in the CRELD1 gene, one inherited from the mother and one from the father. Because our lab had previously studied CRELD1, he reached out to us to test the variants in C. elegans. The ease of genetic manipulation of C. elegans is especially useful to reproduce biallelic variants in which each allele of the same gene carries different changes, just like in the child genome. As a result, we were able to investigate how the two variants together affect CRELD1 function.
What do the experiment in C. elegans show?
Individually, one variant caused mild effect, and the other led to complete loss of function. But, in the context of the biallelic variant, like in the patient, the effects were severe. CRELD1 failed to localize properly. Muscle acetylcholine receptors were dramatically reduced, and the worms showed locomotor defects. This provided clear evidence that the variants together are pathogenic.
You also performed experiment in Drosophila. What did you find?
Since CRELD1 loss impaired movement in C. elegans, we asked whether this function is conserved. In collaboration with Margaret Buller, we found that the locomotion was also impaired in Drosophila when CRELD function was disrupted.
In conclusion, what is the main message of this work?
With our work, we provide an original way to test the impact of human variants using C. elegans, especially in the context of heterozygous biallelic variants. Specifically, we modeled in C. elegans the missense and the truncated variants inherited by the patient from each parent, and demonstrated that the specific combination of these two variants carried on different alleles of the gene CRELD1 is pathogenic. It impairs CRELD1 protein localization, the levels of muscle acetylcholine receptors, and locomotion.
Thank you, Manuela, for this work.
Thank you.